Hi everyone, this is Dr. Jyoti Bala. I welcome you all on my YouTube channel. In this session, we will be exploring some of the useful lab experiments which is required for structure and binding studies. As we know, studying the structure and the binding of biomolecule is crucial for understanding their function and potential application in various fields such as medicine, biotechnology, and biochemistry. Here are a few of the wet lab experiments commonly used for studying the structure and binding of biomolecules. The number one is X-Ray Crystallography. This determines the three-dimensional structure of biomolecules such as protein and nucleic acid. X-ray crystallography crystallizes the biomolecule, exposes the crystal to X-rays and analyzes the diffraction pattern to determine the electron density. The next one is NMR Spectroscopy. This is nuclear magnetic resonance spectroscopy. This analyzes the structure, dynamic, and the interaction of biomolecule in solution. Usually, this involves dissolving the biomolecule in suitable solvent, applying a magnetic field, and measuring the resonance frequency to study the structural information. Chromatin immunoprecipitation, the chip assay. This involves in investigation the protein-DNA interaction and identifying the binding site of specific protein on DNA. It involves the cross-linking and sharing the chromatin, immunoprecipitating the protein of interest with the specific antibody, and analyzing the DNA by PCR or sequencing. The next significant technology is surface plasmon resonance that is involved in studying the biomolecular interaction in real time, including protein-protein and protein-ligand interactions. Usually here, you need to immobilize your binding partner on the sensor surface and allow the flow to the other binding partner. And thus, you can monitor the changes in the refractive index based on the binding. The next one is ITC, isothermal titration calorimetry. This quantifies the biomolecular interaction, including the binding affinities and thermodynamic parameters. Here you need to titrate one biomolecule into the another while measuring the heat release or absorbed during the binding processes. The next technique is mass spectrometry, which determines the molecular weight and identifying the post-translation modification of protein and other biomolecules. The step usually involves ionizing the biomolecule, separating the ion based on the mass to charge ratio, and detecting and analyzing the result mass spectra. The next one is circular dichorism. This analyzes the secondary structure of protein, nucleic acid, and other biomolecules. This usually measures the differential absorption of left and right circularly polarized light to determine the secondary structure's content. Adding on the list, the next technique is electron microscopy, which visualizes the ultrastructure and morphology of biomolecules and complexes at the higher resolution. Usually, this technique involves preparation of sample and then applying the electron-dense strings and using electron beams to visualize the sample structure and interaction. These experiments collectively offer comprehensive approaches to understand the structure and binding properties of biomolecules, enabling the researcher to unlock their potential application in various scientific and medical fields. I hope you have liked the session. Don't forget to like and subscribe the channel, and do share these information among your scientific endeavor. Thank you.